All right, so we're going to end the first day of week 2 by talking about aerobic metabolism. Okay, and so here are the things that we're going to talk about. So really we're just talking about the Krebs cycle and the electron transport chain. Okay, so the Krebs cycle is kind of the intermediary between glycolysis and the electron transport chain. We do produce a little bit of ATP, but what's really important, at least in my opinion, about the Krebs cycle is that we produce a lot of other things. Okay, so we produce a lot of NADH and a lot of FADH2, and both of those things will go and be utilized in the electron transport chain so we can produce more ATP. All right, now this occurs in the mitochondrial matrix. Now this has nothing to do with Keanu Reeves, and I know it's a lame joke that I use every semester, but there you go. All right, so the mitochondrial matrix is basically the fluid inside the mitochondria. Now, the Krebs cycle actually starts with acetyl-CoA. Okay, pyruvate, if you look here, there's no pyruvate in this cycle. There's 8 different steps, okay? And so what we have to do is we have to take pyruvate and we ultimately convert it into acetyl-CoA. And that's called the linking step, and we're not going to focus on that, but I just at least want to make you aware of it. All right, now if you recall from glycolysis, 1 molecule of glucose produces 2 pyruvate. So each of these pyruvate will go through a full turn of the Krebs cycle, and by doing this we produce 1 ATP. So pyruvate 1 goes in, we create an ATP down here at the bottom, and then the other pyruvate can go through and it creates another ATP. All right, so it doesn't technically require oxygen, but like I said earlier, and I said it on the last video and I said it here already, without oxygen we don't produce pyruvate, and if we don't produce pyruvate we can't produce acetyl-CoA. Okay, instead we produce lactate, and lactate does its own thing. All right, and we produce some NADH and some FADH2 as well. All right, so just like the ATP-PCR system, ATP-PCR system had creatine kinase, and glycolysis had phosphofructokinase. Well, the Krebs cycle has an enzyme called isocitrate dehydrogenase, and that's the rate-limiting enzyme. Okay, it's the one that's kind of most sensitive. It makes the biggest deal. So it's responsible for the 3rd step, just like phosphofructokinase. I won't ask you that on a test. You're not required to know that. It's just kind of interesting to me that they're both responsible for that 3rd step. Just like phosphofructokinase and creatine kinase, it's sensitive to ATP, ADP, and phosphate concentrations. If I have a lot of ATP floating around, isocitrate dehydrogenase doesn't do a whole lot. But if I have lots of ADP floating around, isocitrate dehydrogenase does. And again, just to reiterate, this is a negative feedback loop. Okay? And so that's really the primary things that I want you to know about Krebs. And then we go from Krebs into the electron transport chain. Now I also want to make clear that it's not like we go glycolysis, Krebs, electron transport. All of these things are going on all the time because we've got the products. We've got the required molecules, or we can have the required molecules. So all of these things are going on at once. Now the electron transport chain occurs, like it says, on the inner membrane of the mitochondria. So in this little figure that we've got here, we've got the inner membrane and we've got the outer membrane. Okay, and so what happens is different things move across the inner membrane. What happens is those molecules of NADH and FADH2 carry electrons. Okay, makes sense? It's an electron transport chain. And so the electron transport chain, just simply put, is a series of reactions. And through these reactions, we can ultimately create ATP. Okay, so each reaction moves electrons from one place to another. And as electrons move, energy is released. And so we need to harness that energy. Okay. And this is the reason why oxygen is so important, because oxygen will hold on to those electrons, okay? And it actually combines with hydrogen ion to form water. And you can see that, I can't find my cursor. There it is up there. Sorry about that. So right here, we use oxygen to pick up electrons, and ultimately we form water. So as you just live, you're actually creating water, forming water in your body, okay? But perhaps more importantly, we're producing ATP, okay? And so that's how the electron transport chain works. We're just transferring electrons and we're forming water. Okay, and we're going to keep it kind of that simple for here. Okay, we're not going to get into each of the steps, that sort of thing, but just kind of take it. We're going to keep it basic because it gets pretty complicated. If you're interested in learning more, that's what biochem is for. Okay, and so what happens is we take this NADH and it can form about 3 molecules of ATP. You might actually see it as 2.5 in textbooks because it's not exact. It's kind of an average. So anywhere from 2.5 to 3 molecules, 1 molecule of FADH2 produces 2 molecules of ATP. Okay, if we have lots of ADP, electron transport chain is on. If we have lots of ATP, electron transport chain kind of slows down. Okay, and so what's happening is we're doing oxidative phosphorylation. That's another name for the electron transport chain. The electrons that are taken away from NADH and FADH2 go through a series of carriers to produce ATP. And this is just the same thing that I've already told you. I'm just telling you it in a different way. Okay, so we can produce anywhere from 2.5 to 3 ATP or 1.5 to 2 ATP for FADH2. We take the hydrogen ions from each of these molecules and they combine with water or combine with oxygen to form water. Okay. Sorry, there we go. So we're pumping hydrogen ions across the inner membrane. This creates a gradient. Everything comes back to gradients. I love gradients. Okay, and so if we've got a big gradient, well, there's going to be some energy kind of stored in there. And so as we release this energy, ATP is formed as the hydrogen ions go back across the membrane. Okay, so what you see here, hydrogen ions being pumped. And if we're pumping, that means we're going against the gradient. So we're going to have lots of hydrogen up here, not so much down here. And then ultimately what happens is the hydrogen ions diffuse down. There's energy that's released when this happens. And we take that energy to combine ADP and phosphate, and we create an ATP molecule. Okay, and that's how we're going to end with aerobic metabolism.